And I'm going to bring him on now. Daryl Dodson is the Vice President and General Counsel of AIkido Pharma, ticker AIKI, also on the NASDAQ. Daryl is here. Daryl, good afternoon.
Good afternoon, everybody. How are you?
Doing okay.
I'm attempting to share the screen. I had a little trouble there, but I think I'm getting there.
I think I see it. Can you go in to
full screen? Okay.
Yep, full screen.
Okay. Is that it? That look good?
That's it. Looks good.
Okay. Well, hi, everybody. My name is Daryl Dodson. I'm Vice President and General Counsel of AIkido Pharma Inc. And I'm here today to tell you about our company and just highlight 3 of our programs that we have going on since we have a short time. That's our safe harbor statement. Our company objectives are to develop drugs to a stage of a profitable value inflection point. And our strategy for doing that is to co-develop drugs starting in as late a clinical stage as possible. So our technical strategy is preferably we've got drugs that are already in human trials, or if they're not in human trials yet, the IND is already submitted, or if the IND hasn't been submitted, they're at least in the animal studies, the pre-IND animal studies. We want to see robust animal data in the literature and peer-reviewed journals. Usually it's mice or rats. And very importantly, we want to see strong IP filings and hopefully issued patents, preferably issued patents on the drugs themselves before we enter into a kind of a relationship with another party on a piece of technology. We have a very strong financial position right now. It's the strongest position that we have been in since we started. I've been with the company since 2013. It's the strongest financial position we've had since then. We have over 100 million in cash equivalents, and marketable securities, and we have less than $7 million burn yearly. Our stock is currently trading below cash. We have no pending registration statements. We just withdrew the registration we did have pending, so there'll be no dilution in the near future. There'll be no cash raises in the near future. We have no debt, and we have no unusual financing. No preferred shares out there where people have some crazy ratio that they can convert. So these are the 3 investment highlights that I want to tell you about. The first is a prostate cancer treatment that's being developed at Weill Cornell Medical College. It is a dual drug radiotherapy. That means it's got 2 radioactive drugs in it. Prostate cancer is a very, very serious cancer. It's the most common cancer in men. In 2021, it's estimated there'll be over 248,000 new cases and over 34,000 new deaths. And cancer is the 2nd biggest killer in the country. So this is a very, very important and serious problem for men. The global market in 2019 was $7.22 billion, and it's estimated to reach $10.67 billion by 2027. The second program I want to tell you about is a pancreatic cancer drug called, variously in the literature, it's called DHA DFDC or GIM-DHA. I'm just going to call it GIM-DHA because it's easier to say. And we're developing that for oral administration. That comes out of the University of Texas at Austin. Pancreatic cancer is also a very serious cancer. It's difficult to detect early. There's a very low survival in the first 5 years. So it really has unmet need in that field. In 2021, the estimated new cases are 60,000 new cases, over 60,000, and over 48,000 new deaths. So it has a pretty high death rate, like I said. The global market in 2020 was $2.41 billion, and it's estimated to be $3.47 billion by 2025. And switching to antiviral therapies, the third program I want to talk about is what we have from University of Maryland. It's a pan-viral drug design, which means that it treats multiple viral infections. It's effective against the coronaviruses, the serious ones we know, SARS, MERS, and the current one that causes COVID-19. It's effective against flu, Ebola, and others like Marburg. The global flu market alone was projected to reach almost $1 billion by 2026. So let's launch into this dual radiotherapy for prostate cancer. And I just want to tell you at this point, if you want to see anything in more detail, we have a pitch deck on our website that covers our projects in more detail. So I welcome you to go to our website at aikidopharma.com and take a look at that pitch deck. And on our investor page are our SEC filings as well. So I'm referring to one of those here. We have an ownership interest in a company called Convergent Therapeutics, which is licensed by and working with Weill Cornell Medicine on some human clinical trials that are going on right now. So this is a drug that is in human, so it's well advanced. And if you want details on that deal, you can look at our Form 8K that was filed on February 3rd. That's there on our investor page. This was developed under the direction of Dr. Neil Bander at Weill Cornell Medicine, and he's also the lead inventor. You'll see that name again. The treatment contains 2 different radioactive drugs. The first is a radioactive antibody, and the name for it is either actinium-225AC, actinium J591, or it's called Convo-1 alpha, and it has actinium attached to the antibody. The other molecule is a small molecule called lutetium PSMA INT. And INT stands for imaging and I forgot. I'm sorry, but imaging and testing, I think, and it's used ex-US for imaging in patients with cancer using lutetium. The clinical trials right now that are going on are led by Dr. Scott Tagawa at Weill Cornell Medicine. They have just finished a phase 1/2A clinical trial on the antibody actinium J591, and that data is about to be presented here in this quarter, 2Q 2021. They also have an ongoing phase 1B/2A multiple ascending dose trial with that antibody, the actinium antibody, and they were just approved to start the dual therapy trials using both the antibody and the radioactive small molecule, and that's been approved by the FDA. And they're also pursuing additional human trials on the dual drug. Just want to show you a cartoon of how this works. So the green represents the antibody, and the little star beside it represents the radioactivity. And the yellow triangle represents the small molecule, and it has a star next to it representing the lutetium, the radioactivity. So this is a cartoon of a tumor cell, and the red line represents the membrane of the tumor cell. On the tumor cell, on prostate cancer tumor cells, there's a receptor called PSMA. It stands for prostate-specific membrane antigen. And these 2 drugs bind specifically to this receptor. They bind in different spots on the receptor, so they don't compete with each other. So that makes them able to bind to the receptor at the same time. A very cool feature of this drug is that the antibody causes the PSMA receptor to be internalized. And when that gets internalized, both of the drugs that are bound to the PSMA receptor are basically injected in cells. So you get an injection of 2 different radioactive drugs into the tumor cells. These 2 drugs, when they're injected into patients, go to different parts of the body. They go to different tissues in the body. That's what non-overlapping biodistributions means. And what that does is it's a very important feature where it reduces the potential for additive damage to healthy tissues. And that's a concern anytime you're using radioactivity in patients. So just to give you a few success data points on radiotherapy, and these all have to do with Novartis. In October 2017, Novartis acquired a drug called Lutathera, a lutetium dotatate, for $3.9 billion. And then a year later, in October 18, they did a $2.1 billion license with Endocyte to acquire lutetium PSMA 617. That is their, as far as I can tell, that's their current frontline drug on this, because they just came out with, and this is just over a month ago, they came out with positive phase 3 data on this drug, lutetium PSMA 617. And that's an actual image from the title from Novartis' website where they announced it. The relevance for us is that our lutetium PSMA INT, or Cornell's PSMA INT, for the dual therapy is very similar chemically, structurally, to the Novartis drug. It has the same PSMA binding structure on one side of the molecule, and it has the same lutetium binding structure on the other. And obviously, they both bind to lutetium. So the take-home message here is we've got data coming out very soon on the antibody portion of this dual therapy, and we've got some very good phase 3 data on a very similar molecule on the small molecule of the dual therapy. So I want to go into our second program that we're involved in. We're licensed from the University of Texas Austin for this GEM-DHA for pancreatic cancer. We're licensed to both the drug and to a potentially next generation oral delivery system. There's 2 different IP portfolios for those 2 different things. And just to direct your attention to the cartoon on the side, it's a very overly simplistic rendition of the chemistry here, but I just wanted to do it for understanding. Gemcitabine is a common and already FDA approved chemotherapy drug that's used for like 5 different cancers. And our drug is a modified version of gemcitabine, where it's chemically attached to the gemcitabine is an omega-3 fatty acid called DHA. So that results in this, what I'm calling GEM-DHA. And then here we go into the oral formulation portion of it. That is mixed with a blend of different kinds of lipids, and then those form solid lipid nanoparticles. The data in mice show that those solid lipid nanoparticles allow it to be administered orally, and it allows it to cross through the stomach acid, through the stomach, and get into the blood so it can go to its target. Like I said, it has really good data in mice. There's 4 different publications that are out on this. They're listed in our pitch deck. So again, I encourage you to go take a look at that. This drug, it was unexpected at the beginning, but it turns out of all organs, it concentrates itself in the pancreas. So we think it's going to be great, have great potential for pancreatic cancer. That's why we're pursuing it in that indication. It has much higher activity than gemcitabine. In fact, in cells, GEM-DHA requires 100,000 fold less drug to treat the cells than the tumor cells than does gemcitabine. It has a promising safety profile in the mice, and it greatly reduces drug resistance that you see with other types of chemotherapies. Our R&D is progressing on this. We've already produced product at high yield. Our manufacturer has replicated the procedures in the literature and already produced a procedure that produces this at high yield, and the scale-up is currently being validated, and batch-to-batch testing is underway for purity and consistency. Our next steps are going into formulation development and cGMP manufacturing protocol, and we'll be hiring a larger organization for that, and we'll be announcing that. And we'll also be hiring a animal studies outfit for our pre-IND studies, and we'll be announcing that as well. And the last study is a computer design antiviral treatment. This is the Panviral Treatment I talked about. It was designed using computer learning, computer-assisted modeling. We have an exclusive license, and we paid for a sponsored research agreement to keep the research ongoing in-house at UMB with the inventors who invented it. So we're collaborating right now with UMB to optimize these licensed candidates. And the cartoon over to the right is a little complicated, but basically, just to show you that the drug targets what's called the SCI complex. The SCI complex is made up of multiple proteins. And when those proteins assemble together, they make the SCI complex, they form a substrate for RNA turnover. And these scientists discovered this, that SCI complex and its ability for this RNA turnover is very also important for the viral replication of all these different viruses. So what they did was they did computer modeling on the structure of this SCI complex. They identified pockets on the SCI complex where drugs might bind. This is by computer modeling. And then they designed drugs by computer modeling. And then they actually synthesized the drugs that they had modeled. So they don't know anything yet about the drugs. And they test them. And lo and behold, they actually there's lead candidates that have really good activity against all these viruses, again, including flu, all these coronaviruses, and Ebola and Marburg. So right now, the structures are being optimized to get just the very best structure that we can then go forward with into the future. So that's the third project. I want to show you some names that are on our scientific advisory board. These are names you've already seen. Dr. Scott Tagawa is on our scientific advisory board. He is the one leading those clinical trials in the dual radiotherapy that Weill Cornell's doing. Dr. Neil Bander is the inventor of that dual therapy, and he is actively involved. Dr. Rong Kui is the lead inventor of both the drug from UT and the oral formulation from UT. And Dr. Matthew Freeman, who I just mentioned, is the inventor and the lead scientist who's continuing the research on the Panviral therapy. And these guys are on our scientific advisory board. They talk to us a lot. They've been very helpful in our development so far, and I think it's going to really give us an advantage to have these relationships going into the future. So that's the presentation. This is our company contacts. Anthony Hayes is our CEO. There's the telephone number for the company and the website. And Brett Moss, our managing director of investor relations, is Hayden IR. There's his phone number. If you have any questions for investor relations, you should give him a call. So thank you very much for attending. Do we have time for questions?
We do have time for questions, and we have a couple questions actually. And I'm bullish because University of Maryland produced this guy, and so go Terps. All right, let's go to some questions from the chat here. Let's see, we have a few. How long will the UMD research take to conclude?
Well, the UMB research is pretty early right now. It's actually in cell culture that they're doing this. We think that the lead candidate for moving forward with that is going to be done within the next 6 months. We have milestones, or 6 to 12 months. We have milestones in our agreement for that. But at that point, we'll have a lead candidate then to go forward with, to go into animals.
Here's a question from DB. How does the effectiveness of your pan-viral drug compare to the effectiveness of competing drugs that target specific viri or viruses?
Well, I mean, these drugs work down to the nanomolar levels, so they work at very low physiological concentrations. I can't tell you just sitting here what how the dosages are different, if that's the question. But they're extremely effective against the viruses at the dosages that are effective. So,
okay. I think that was all as far as questions. Let me double check. Yeah. Okay. Daryl Dodson, Vice President, General Counsel of AIkido Pharma. It's up on the screen. The ticker is AIKI. The website is right there in front of you. Daryl, thank you so much for stopping by, and a great way to wrap up the first day of the Benzinga Global Small Cap Conference. Thanks a lot.
Thank you very much.